Let's look at RNA. So RNA stands for ribonucleic acid. Okay, all RNA now in a current form comes from DNA, but RNA was believed to be the precursor or the original nucleic acid that was reversed into DNA, reversed to DNA because DNA was a more stable form of storage. But RNA, by most accounts, came before DNA in our evolution and our development. Okay, across all species, not just humans. All right. So we also have then there are three types of RNA. So all of these RNA that I'm about to discuss with you are made from DNA now, okay? And they are usually made inside the nucleus. So the first one, so types, well first, before we even do that, the RNA nucleotide, right here, again, nucleotide is nucleotide. So here, but this time the sugar in RNA is ribose, okay? And of course, attached to number 1, you have bases. Attached to number 5, you have a phosphate group, okay? So the potential bases are, for RNA, there are adenine, guanine, cytosine, and uracil. There is no thymine in RNA. It's just uracil. So again, so you'll have A, G, C, U as potential bases. Okay, now RNA. So again, back to the types of RNA. Okay, so RNA types. You have messenger RNA, mRNA. This form of RNA, again, is made from DNA in the nucleus. So it's made in the nucleus. From DNA, okay? And this mRNA carries the codes to make proteins. This is the mRNA this is the RNA that comes from our genes. Our genes is DNA. When we copy those genes over to make proteins, we make that DNA into mRNA. So mRNA carries the codes from our genes that tell us how to make our proteins, meaning how to assemble the amino acids together. We also have, that's one type. Another type of RNA is your rRNA, small r, which stands for ribosomal RNA, made in the nucleolus, which is a part of the nucleus, again from DNA. And RNA acts like an enzyme. It's actually an enzyme that's involved, it works inside the ribosome, and it is involved in forming peptide bonds between amino acids. That's rRNA, okay? So normally function is the purview or the business of proteins. But here's a case where RNA is doing function. It's functionally, it's acting as an enzyme, even though it's not a protein. So this is why RNA is believed to be an earlier nucleic acid before DNA, because it can do stuff. And then we have the third type of RNA is your transfer RNA. tRNA made in the nucleus from DNA, and And this tRNA, its job is to bind to and transport amino acids around the cell. Okay, so you have three types of RNA. mRNA carries the codes to make proteins. rRNA helps to form the bonds in amino acids between proteins. And tRNA, between amino acids, and tRNA transports the amino acids into the ribosome to be assembled into proteins. So all three of them are involved in protein assembly at some protein synthesis at some point. That's a big word for me there. Okay, then we go to something called the genetic code. So RNA forms the genetic code, and the genetic code is a series, or refers to triplets or triples of RNA nucleotides that code for amino acids. All right, and they're called codons. They're called codons. Okay, these are the triplets. So maybe something like A. That's a codon. C, codon. CUA, codon. Okay, RNA triplets are called codons. And since you have 4 possible codons to be arranged in triplets, the possibilities are 4 to the third, so which means you have 64 possible codons. 64, okay? And these 64 possible codons code for 20 amino acids, which means they must have multiple, you must have repeats of codons, maybe 3 or 4 codons code for the same amino acid. Otherwise, you will only have 20, right? So there's some redundancy there in your codon structure. Of the 64 codons, I want to know 4 of them, okay? So there's a codon called AUG. That's called the start codon, meaning all proteins, when they're being made, the very first codon that is translated over is AUG. And it codes for the amino acid called methionine. So the methionine amino acid is the first amino acid in all proteins. It may be cut off later when it's done, but in making it, this is the first one you make, that you bring into the chain. Methionine from the start codon. And then there are 3 stop codons, meaning when the ribosome gets to any of these 3 codons, it stops making the RNA, making the protein. And they are UAA, UGA, and UAG. These are called your stop codons. When you see them, you stop making the protein. All right, so that's RNA and the genetic code. We also have other forms of nucleic acids which do different things. Among them is, of course, a very famous one, which is ATP, right? It's also a nucleic acid. So ATP, which stands for Adenosine Triphosphate, okay? It's formed by, again, you have ribose sugar. It's basically an RNA nucleotide. You have your ribose sugar there. And of course, attached to number 1 is your base. In this case, it's adenine base. It's attached to there. And then attached to number 5, carbon, are 3 phosphates. 1, 2, 3. So that's called adenosine triphosphate. 3 phosphates there. And these are high energy bonds. When they break, they release energy that's used to do work inside the cell. And when you take off one of them and you're left with, say, this one, with just 2 phosphates there. And this is called ADP, adenosine diphosphate. And then, so this is ATP. It has 3 phosphates attached to it. And then you can still further break down the remaining diphosphate by removing one more phosphate group, and what you're left with is AMP, adenosine monophosphate. These are all nucleotides, RNA nucleotides that's used for energy. Okay, we'll stop there.